Hello, my name is Richie. I am part of BCU's Master's Environmental Studies program. Today, I'll be talking about what drives productivity in riverine rock pools. In aquatic systems, we have productivity occurring, which is a process of inorganic carbon fixation within an ecosystem. GPP, or gross primary production, is the total amount of inorganic carbon being fixated. In other words, the process of photosynthesis. What drives GPPs varies throughout different aquatic systems. For example, the amount of sunlight is a main influencer of GPP in rivers. In vernal pools, we see phosphorus and nitrogen driving GPP. In lakes, we have seen some suggestions of allogenous inputs or external sources of carbon such as sticks, leaves, and seeds influencing GPP. With rock pools being influenced by rivers and having characteristics of very small lakes or vernal pools, we hypothesize that shading, temperature, and allogenous inputs are the main drivers of GPP in riverine rock pools. With the numerous variations in the rock pools, we want to determine if these variables or others drive GPP. Some of the variations that we are examining will be shading, distance from forest edge, volume, temperature, depth of the pool, coarse benthic matter, fine benthic matter, total benthic matter C and H concentrations and its ratio. We also see variabilities in dissolved oxygen while collecting it to calculate GPP. We collected data from 27 pools on the north side of Belle Isle. For shading, we measured this using a densitometer that is positioned 12 inches above the pool's water surface. Depth was measured using a measuring stick from the bottom of the pool to the pool's lip. ArcGIS was used to measure the distance of each pool to the forest edge. The volume was quantified by using 2 liters plastic containers to empty out the pools and stored in plastic bins. After emptying the pools, leaf litter and sticks were collected from the pool to determine coarse benthic matter. A water sample was collected from the plastic bins and were used to calculate fine benthic matter and chlorophyll A. Coarse and fine benthic matter samples were ran using a CHN analyzer to obtain carbon and nitrogen concentrations. With this in hand, we calculated total benthic matter and the total benthic matter C to N ratio. As mentioned earlier, we also collected dissolved oxygen data and temperature with our sondes. The sondes were positioned 5 centimeters below the water surface and left for a period of 5 days. Here are a few histograms of the data that was collected. For temperature, this is in degrees Celsius and is an average over the 5-day period. Shading is a percentage of the pool that was covered by tree canopy. These histograms show that these pools do have characteristics that vary from one pool to another. Dissolved oxygen also had a lot of variability as well. The top graph shows a diel curve, which represents the cycle of carbon being fixated and consumed. The black line represents the amount of dissolved oxygen in milligrams per liter, and the blue line shows the temperature in degrees Fahrenheit. When the blue and the black line are increasing in the top graph, it signifies that there are more GPP occurring than respiration or the consumption of organic carbon. As it decreases, the opposite is true. For the bottom graph, the blue and black line represents the same thing. However, as you see, there is no diel curve and the temperature is fluctuating a lot less than the top graph. This shows that this pool isn't going through the same processes of productions and consumptions of carbon as a pool that is represented by the top graph. Each graph outputs a raw data set that gives us the temperature, dissolved oxygen saturation, and dissolved oxygen in milligrams per liter at 15-minute intervals. With this, we calculated where dissolved oxygen would be fully saturated or at equilibrium using the temperature. We are doing this because our rock pools are a smaller aquatic system than compared to lakes or rivers. Using the dissolved oxygen in milligrams per liter, we divided that by the calculated dissolved oxygen equilibrium to get a new dissolved oxygen saturation. This is then compiled and separated into days. The dissolved oxygen saturation in milligrams per liter is then averaged for every 30 minutes. Atmospheric exchange is calculated using average 30 minutes dissolved oxygen saturation. Next, the dissolved oxygen flux is calculated. The flux determines whether the pool is producing or consuming more dissolved oxygen, therefore carbon. To calculate GPP, which is again the amount of inorganic carbon being fixated, we have to first determine net ecosystem production and ecosystem respiration. To do so, we have to find the hours of daylight or when sunrise and sunset is for each day. We first calculate net ecosystem production, or NEP, which is the rate of dissolved oxygen production during the day with respiration occurring. We do this by adding up all the dissolved oxygen flux in the daytime. Then ER, or ecosystem respiration, is determined by adding up the amount of dissolved oxygen flux at night. This will give us how much respiration is occurring only at nighttime. That number is then divided by how much nighttime there was to obtain a rate of respiration that is occurring per hour. We then take our NEP and the absolute value of ER by the amount of daylight to determine the rate of GBP for a single day. This is repeated over 4 different days and average. As you can see, there is also differences of GBP within our pools. After log transforming some of our other variables, we did a multiple regression with shade, temperature, log volume, TBM ratio, log chlorophyll A, log TBM, and log distance. We see that only temperature was correlated with log GBP. We then try to simplify the model by running a stepwise AIC. The model only retained temperature, log volume, TBM ratio, and log distance as the most important variables and explaining 66% of the variability in log GBP. However, only the variables temperature and TBM ratio seems to be significantly correlated, while log volume seems to be only marginally significant. Looking at the univariate relationships of those variables from the models, we see their individual relationships. With temperature and volume, you see positive relationship with log GBP, while with log TBM ratio, we see a negative relationship with log GBP. In conclusion, only one of my hypothesized variables, which was temperature, seemed to be significantly correlated with log GBP. Our model also predicted volume and TBM ratio as being correlated with log GBP as well. I would like to thank everyone in Vanish Lab and Dr. Bukavakis for their help.